Hi everyone, my name is Laura, and the title of my research project is Investigating the Association Between Prenatal Stress and Neurodevelopment, the Contribution of the Gut Microbiota. To begin, approximately 30% of pregnant individuals worldwide report significant stress during pregnancy. Prenatal stress is associated with adverse neurodevelopmental outcomes in children. For this reason, researchers in the field of maternal and child health want to understand how prenatal stress is transferred from the mother to the baby and how stress can program the fetal brain. Emerging evidence suggests that the gut microbiota may mediate the relationship between prenatal stress and neural development. The gut microbiota are all the bacteria that are present within the gastrointestinal tract. Bacteria within the gut are essential to human health because they can produce metabolites like short-chain fatty acids and neurotransmitters that the central nervous system can sense. Both human and animal studies have linked the gut microbiota to cognitive and mental health outcomes. With respect to neurodevelopment, the argument that is being made is that the type of bacteria that first colonize the infant gut matters because particular bacteria will produce specific metabolites that the brain will sense during a critical window of development. Prenatal stress has been associated with the child's gut microbiota composition. And for this reason, the objectives of the study are to first determine if features of the child's gut microbiota are different depending on the amount of prenatal stress exposure, and second, to determine if children's full-scale intelligence quotient and primary indices scores are different depending on features of the child's gut microbiota. I used data collected from the Alberta Pregnancy Outcomes and Nutrition Study to answer both objectives. I operationalized prenatal stress using maternal salivary cortisol samples and mood questionnaires. Specifically, I used the Edinburgh Postnatal Depression Scale and the anxiety subtest of the Symptom Checklist 90 to generate a psychological distress score at each trimester. Child stool samples were collected at the age of 3 to 4 and were used to quantify gut bacteria using 16S rRNA sequencing. Lastly, the Wechsler School and Primary Scale of Intelligence was used for neurocognitive assessments. I want to draw your attention to Figure 1, a correlation heat map examining the relationship between maternal stress measures at each trimester with child's alpha diversity measures. Alpha diversity is an index that lets us know the number of different bacterial species found within the gut. In other words, this index is a macroscopic index that allows us to understand how diverse the gut microbiota composition is. The blue indicates a positive association between variables, and the red indicates a negative association. I want to highlight some significant trends. Let's look at the left side of the graph. We see that cortisol in trimester 1 has a significant positive association with alpha diversity. In contrast, cortisol has a significant negative association with alpha diversity in trimester 2. In comparison, if we look at the right side of the graph, we see that the psychological distress score in trimester 2 has a significant negative association with alpha diversity. These results indicate trimester-dependent effects and suggest that maternal cortisol and psychological distress relate differently to the child's alpha diversity. I want to now draw your attention to Figure 2, a correlation heat map examining the relationship between maternal cortisol measures and taxa abundance at each trimester. The critical trend I want to highlight here is that maternal cortisol exposure is significantly associated with taxa abundance in trimesters 1 and 2. As a next step, I wanted to explore the relationship between the psychological distress score and taxa abundance at each trimester, as I had done with cortisol. I want to draw your attention now to Figure 3. I found that the psychological distress score in trimester 2 is positively associated with ruminococci, as seen by the correlation plot. Collectively, figures 1 to 3 indicate that prenatal stress is associated with features of the child's gut microbiota at the age of 3 to 4. However, it is essential to now explore how the gut microbiota features relate to cognitive outcomes. I want to draw your attention to Figure 4, a correlation heat map for the relationship between taxa abundance and full-scale IQ in the primary indices. What I found was that visual, spatial, and the working memory composite score have a significant positive association with the abundance of particular taxa. To summarize, the maternal cortisol concentration and the psychological distress score are significantly associated with the child's alpha diversity and particular taxa abundance at the age of 3 to 4 in a trimester-specific manner. In addition, bacterial taxa abundance is associated with visual, spatial, and working memory composite score. To conclude, these findings are important because previous research suggests that differences in microbiota composition are associated with differences in cognitive outcomes in early childhood.